Welcome to The Explainer. Today, we're diving into a pretty startling new frontier in pollution science, the discovery of microscopic plastics inside the human body. We're going to break down the progress, the big problems, and what's coming next in this really fast-moving field. So, let's just start with a question that until very, very recently felt like it was straight out of science fiction. But as you're about to see, it's becoming a very real scientific question. And here's what we know right now. A huge review of studies confirmed that scientists have found these tiny plastic particles in at least 15 different parts of our bodies. And we're talking about places like our blood, our lungs, and even in the placenta, which suggests this exposure starts before we're even born. Okay, let's get into it. Here's our game plan for today. We'll start with what scientists have found so far. Then we'll put on our detective hats and see how they're actually finding these particles. After that, we'll tackle the biggest challenge in this field, contamination. And finally, we'll look ahead to see where all this research is going. All right, first up, the plastic inside us. Let's really try to get a handle on just how widespread this whole thing is. Now, what's really interesting is where the research has been focused. You can see here that about 30% of studies looked at feces. And the reason for that is pretty simple, It's a non-invasive way to see what's passing through our system. But as you can see, they're also looking in much tougher places, like our lungs and our blood. And get this field is moving at an incredible speed. The very first paper on this only came out in 2019. A whopping 80% of all the research has been published in just the last couple of years. It is a scientific sprint, and it's quickly spreading across the globe. So, this all raises a huge question, right? How on earth do you find these tiny, invisible invaders? Well, this is where the real scientific detective work comes in. Okay, the first crucial step is something called sample digestion. And the best way to think about it is like trying to find a single plastic needle in a giant haystack. But instead of digging through all the hay, you just dissolve the entire haystack, leaving only the needle behind. And that's basically what scientists have to do with human tissue to get to the plastic. It's this really meticulous four-step process. First, you have to collect the sample with extreme care. Then you do that digestion process we just talked about. After that, you filter what's left to catch any particles. And finally, you use these high-tech tools like Raman spectroscopy, which is kind of like a chemical fingerprint scanner, to prove that a particle is actually plastic and not something else. And this shot just shows you how complicated this all is. To get good results, scientists need this whole shopping list of very specific chemicals, special filters, and really advanced machines. Every single step is designed to get them a little bit closer to an accurate count of what's really inside a sample. But, and this is a huge but, there's a major plot twist in this detective story. The single biggest problem, the Achilles heel of this research, is that the very thing they're looking for, plastic, is absolutely everywhere, including all over the lab. It's a total contamination minefield. When researchers looked at all the published studies, they found that the safety protocols were all over the place. While most labs are doing basic stuff like cleaning their equipment, some of the most critical steps aren't happening. For instance, less than 20% of studies reported using filtered air. That means airborne plastic fibers from, say, a scientist's clothing could just be falling right into the samples. So what you end up with is this huge gap between the ideal protocol, the dream scenario, and what's actually being reported. In a perfect world, every lab would be this sterile bubble. But in reality, a lot of these crucial measures are used inconsistently. And what's really worrying is that the results from the blank controls, the dummy samples used to check for lab contamination, are often not even reported. And because of this massive challenge, the scientists who wrote this review are basically putting up a big warning sign. They say that we have to interpret the results we're seeing right now with a healthy dose of caution. The risk of contamination is just that real, and it really complicates what the data is telling us. So, with this huge challenge front and center, where does this incredibly important research go from here? What's next? Well, for one thing, we're starting to connect the dots. This table shows some of the most common types of plastic being found. We're seeing polymers from everyday things like plastic bags, food containers, and drink bottles showing up in everything from our blood and lungs to breast milk and the placenta. The path forward is actually becoming much clearer. First and foremost, scientists have to standardize their methods to beat that contamination risk. From there, the focus can really shift to studying vulnerable groups like kids, using huge biobanks to hunt for links between plastic and disease, and starting the search for even smaller and potentially more dangerous nanoplastics. And all of this leaves us with a final, crucial question. This field is really still in its infancy, but it's growing up fast. As scientists get better and better at looking, we have to realize the search has only just begun. And what they find next could redefine our whole understanding of pollution and what it truly means to be living in the plastic age.